Hello, grade 10. Now we will start with section 1 in chapter 8, which is talking about identifying DNA as genetic material. Now, in this lesson, we learned that the DNA was identified as genetic material through a series of experiments. Okay? The first scientist who found out about the DNA, it was called Griffith. He finds the transforming principle. He used two types of bacteria, and one of them is deadly, one of them is not deadly. The S form was deadly, the R form was not deadly, and he injected the mouse. He made the experiment by injecting the mouse with first mouse he injected with the S bacteria, live S bacteria, which is deadly. After a while, the mouse dead. But when he injected the mouse with the R bacteria, which is not harmful, not deadly, the mouse is still alive. After this, he heated the harmful bacteria. He killed it, that means. And after he injected in the mouse, also the mouse is still alive because he killed the S bacteria. But he was surprised when he injected the mouse with R bacteria, live R bacteria, and killed SI bacteria. He was surprised that the dead mouse is dead. And he concluded that there is a transforming material passed from dead S bacteria to live R bacteria, but he didn't know what is this material. So after him, he came Avery and his friends, and they purify all the components of S bacteria. They took all the components, and they found that this transforming material was the DNA, but not all the scientists were convinced by this conclusion. So after Avery, he came Hershey and Chase, and they confirmed that the DNA is the genetic material or the transforming material by tagging a virus, which is a bacteriophage, with DNA, radioactive phosphorus, and another virus of the same type with radioactive sulfur. They observed that the material that injected from the virus to the bacteria was the DNA, not the virus. So they were concluded that this transforming material that infect the bacteria is the DNA. Okay? All right, thank you. Now we will start with the next session اللي هو 8.2. All right, just 1 minute, please. Now in 8.2, we will come to the last section, which is the structure of DNA. Now, after these scientists find out about the DNA, they identify it as a genetic material. Some scientists start to study the structure. Okay, so now DNA mainly it is a nucleic acid. Okay, it is a polymer which is called a nucleic acid, and it is a carbon-based molecule. Now DNA is composed of monomers. These monomers called nucleotides. And each DNA molecule as a polymer made of a nucleotides as monomers. And if you look here at this picture, it's obvious that this is the DNA. DNA, this is a polymer. It is made of different, each one of these is a monomer. Each one of these is called a nucleotide. When they join together in this way, they form the DNA. All right. Now each nucleotide is made of these three parts: a phosphatic group, a deoxyribose sugar, and nitrogen-containing base. Mainly, we have 4 types of nucleotides depending on this part. Okay? This nitrogen-containing base have 4 types. So, depending on these, we have 4 types of nucleotides. It either will be a pyrimidine or purine. Pyrimidines are single ring as thymine and cytosine, or purines as adenine and guanine. All right, so this is the structure of each nucleotide. Now there was a scientist who called Chargaff. He found out by after studying DNA of many organisms, that the thymine and the adenine was equal, have same concentration in the DNA structure. Cytosine and guanine have the same concentration of DNA structure. So Watson and Crick used his findings, and they can model a three-dimensional structure of DNA. Okay, they found out or concluded that the DNA has a double helix. This is very important to know. Has a double helix. Look like this structure. Two strands, double helix structure, and these two double helix are connected by the bases here, adenine, thymine, guanine, cytosine. And what is the bond between them? It's hydrogen bonds. You should know this. Okay, base pairing rules: adenine with thymine, or cytosine with guanine. So don't forget, A with T, C with G. Okay. Now, so in this double-stranded structure, what connect these bases together is the hydrogen bonds. Okay. All right. Now, so after they model the structure of the DNA, okay, they found out also with the help of Franklin, Rosalind Franklin, she found that through the X-rays that a DNA image is like X, okay, double helix. This helped Watson and Crick to figure out the structure of DNA. It's double helix, two strands. Don't forget that A bonds with T and C bonds with G. Now, this is the pair rule. Base pairing rule. Show how nucleotides always pair up in DNA. A pairs with T or C pairs with G. Okay? Because one ring مع two ring. Okay? Now, this is the structure. Now here, this is untwisted. Naturally, the DNA will be double helix. It's like twisted ladder. But if you untwist it, it will look like this. Can you see the hydrogen bonds between the bases? So bases are connected or joined together by hydrogen bonds. Okay. Now, I want to ask you about this question. Okay, this is very important. If you have this sequence, okay, T, G, A, C, T, A, what sequence of bases would pair with the sequence this one? What is the sequence that will pair with this one? T, so the other sequence will be A, G, C, A with T, G with C, and etc. Okay, so I wish you good luck in your exam, and see you too on Sunday, guys. Take care.